Okay, so today we will be starting with Enterobacteriaceae. This is a very important family of bacteria in veterinary microbiology, bacteriology. So let us start. This is the first lecture on Enterobacteriaceae. So Enterobacteria gram negative. These are bacilli. Bacilli means these are rod shaped and length is 3 micrometer. So we need to learn these units that millimeter is used for the um, measurement of the size of fungal and bacterial colony. Micrometer used for the measurement of single cell of bacteria and fungus, and nanometer used in case of viruses, cell of virus, with a single viral particle. Okay, so Enterobacteria gram negative bacilli, length 3 micrometer, and they usually ferment glucose. Usually ferment glucose, but E. coli ferment lactose. Not only E. coli, other examples must be also there which ferment E. coli, not ferment lactose, but E. coli is the most important example which ferment lactose. Okay, next it is catalase positive, oxidase negative. Catalase positive, again it means that it will have an enzyme called catalase which will convert H2O2 into H2O plus O2, that is hydrogen peroxide, into H2 water and oxygen, because hydrogen peroxide is in itself is a free radical. So the bacteria needs to get rid of that H2O2 so that it is not damaged by free radical, because free radical damages in a 3-step fashion. So that is the topic for pathology. But okay. Next is oxidase negative. Oxidase negative, that is, it does not have cytochrome C oxidase, so it will not be able to perform the function which the cytochrome C oxidase can perform. Okay. Now motile enterobacteria have peritracheous flagella. So motile enterobacteria means that and all the enterobacteria which are motile. Not all enterobacteria are motile, but whichever enterobacteria are motile, they are motile because of peritracheous flagella. What is peritracheous flagella? Peritracheous flagella means the flagella present all around the cell, all over the cell of the bacteria. Okay. Now it contains more than 40 genera and 180 species. This family contains more than 40 genera and 180 species. And half of these are of veterinary importance, half of these are of medical importance. Okay. Now Enterobacteria, Enterobacteriaceae can be divided into 3 groups on a very basic basis of classification. That is, whether they cause diseases majorly or opportunistically, or does not cause disease. So on that basis, major pathogen, opportunistic pathogen, and non-pathogen. Okay, so now opportunistic pathogen causes clinical diseases in location other than GIT. Why not in GIT? Because enterobacteria are normal flora of GIT. Sometimes enterobacteria are found as normal flora of GIT. Normal flora means the normal bacterial population found in intestine, which are helpful for or healthy, which are beneficial for that animal. Okay. So next point is that major animal pathogen are E. coli, Salmonella species, and Yersinia species. There must be many more. There are many more, but the most important are these 3: E. coli, Salmonella, Yersinia species. Okay. Now, what is the usual habitat of E. coli? First, we are discussing the general point. So, what is the usual habitat of E. coli? They inhabit the intestinal tract of animals and humans, and whenever present outside the body of animal and human, they contaminate vegetation, soil, and water. Okay. Okay. So next point is differentiation of Enterobacteriaceae. So this means that this heading means that how to differentiate Enterobacteria, of which bacteria to put in Enterobacteriaceae. So any bacteria which is kept in Enterobacteriaceae follows these 5 features. It is oxidase negative, it is a facultative anaerobe, gram negative, bacilli, and grown Maconkey agar. So these 5 characteristics have to be followed by any bacteria who wants to become an enterobacter. Okay. And few of Enterobacteriaceae also produce hemolysis on blood agar. Simple point. So this is not a characteristic of many Enterobacteriaceae. Only few Enterobacteria produce hemolysis on blood agar. Okay. Next point is this. Enterobacteria on the basis of lactose fermentation. So obviously on the basis of lactose fermentation there will be 2 types, and lactose fermenter and non-lactose fermenter. The bacteria which ferment lactose and the bacteria which do not ferment lactose. So the bacteria which ferment lactose are those which produce pink colonies because of acid production from lactose, and colonies which are surrounding colonies of non-lactose fermenter are surrounding medium are pale, and these are alkaline due to utilization of peptones in the medium. Okay. Now colonial morphology, 3 types of colonies are mainly observed in case of Enterobacteria: mucoids forming and red pigment producing colonies. So red pigment producing colonies is produced by a less important opportunistic pathogen, that is called Serratia marcescens. Swarming groups, swarming means making dense groups. So it is produced by Proteus species. And mucoid colonies by Klebsiella and Enterobacter. This point is important. E. coli rarely show mucoid colonies. So this can be asked in true or false. E. coli rarely shows mucoid colonies. Okay.